So our next major pathway for the production of ATP, or I should say the resynthesis of ATP, is via aerobic energy production, or oxidative phosphorylation as we'll call it, using oxygen to phosphorylate ADP into ATP. The first stage is glycolysis, just as we had in the previous video, the breakdown of glucose or glycogen into pyruvate. But now, because we're going to be at an appropriate intensity and oxygen is going to be available, we can continue to break down that pyruvate rather than turn it into lactate, and we can better deal with those pesky hydrogen ions, for want of a better word. So from now on, that pyruvate moves into the mitochondria, specialized cell within the muscles, sometimes referred to in the text as the powerhouse of the cell, because as we'll see as we go on, we get a much larger supply of adenosine triphosphate when we break energy down in this way. So, phases we're going to go through. System's not working, never mind. There we go. So, stage 1 was glycolysis. Stage 2 will be the link reaction. Stage 3, the Krebs cycle. And then the most important one, the electron transport chain, because that's where oxidative phosphorylation is going to occur. The link reaction then is a process by which pyruvate is transformed into a substance called acetyl coenzyme A. So this is catalyzed by an enzyme called pyruvate dehydrogenase. We add coenzyme A, we remove more hydrogen, which can get sent off to the electron transport chain, and we remove some carbon dioxide, which gets shuttled out in the blood and can be breathed out. So that's a fairly simple reaction, fairly small, no energy produced there, but hydrogen is removed, and that's always going to be key for us. NAD, once again, taking that away. Next stage, a Krebs cycle, or sometimes called citric acid cycle. So again, another complex chain of reactions whereby our acetyl coenzyme A that comes in is transformed into a number of other key substrates along the way, and in that process we remove more hydrogen carried by NAD, we create one ATP, we remove more carbon dioxide, and we have another electron carrier called FAD, essentially doing exactly the same job as NAD, being reduced to gain a hydrogen and carrying that to the electron transport chain. The reaction goes on and on. As long as we have new acetyl coenzyme A coming in, it can join with oxaloacetate, which is that final process at stage 10, and we can keep going. That therefore brings us to electron transport chain. This is where we are finally able to deal with all that hydrogen that's being produced along the way, and we're able to neutralize it in a way so it's not going to cause that fatiguing acidosis for us. So hydrogen bonds are really, really important. This is where we're getting key energy to phosphorylate our ADP and create more ATP. So for every NADH that comes to the electron transport chain, the hydrogen is passed down 3 separate receptors until it is finally attached to oxygen to become water, H2O. Every time it's passed on, it releases enough energy to create 1 ATP. So every NAD along the way creates 3 ATPs. So as FADH comes into the transport chain at a slightly later stage, it passes its hydrogen on to only 2 receptors, which therefore means every FADH produced creates 2 ATPs. To kind of summarize the whole process then, if we're starting with glucose, the glycolysis creates 2 ATPs and gives us 2 NADH plus H. Link reaction gives us another 2 NADH plus H. Krebs cycle gives us 2 ATPs, 6 NADH plus H, 2 FADH plus H. And then at stage 4, because all of those NADs pass their hydrogens down to make 3 ATPs each, and the FADs pass their hydrogens down to make 2 more ATPs each, we get a total of 34 ATP created at electron transport chains, giving us a total of 38 ATPs created from one single molecule of glucose. Now obviously this is huge compared to the amount we got from glycolysis alone, where it was 2 or 3, and from phosphocreatine, where we got 1. The downside being, because we're reliant upon oxygen, because we've got such a complex chain of events, the process is much slower and has to be carried out at a much lower intensity.